Hello, my name is Alexander Dematos, and today I want to talk to you about the use of computer models in biological research today. Computer models are applicable in many ways and in many different fields. For example, in healthcare, it can be used for things like tracking infectious disease or predicting drug side effects, all in a manner which saves time and money for researchers. In biology, computers have made it easier to store data in fields like genetics, where tons of information can be stored in these systems and experiments can be completed quickly. Today, I will be focusing on a paper using computational models for genetics. In this study, titled Recombination Drives the Evolution of Mutational Robustness, Dr. Singhal, Dr. Gomez, and Dr. Birch, all of whom work within the UNC system, examine genetic processes and their potential effects on organisms using a computer model. Firstly, I want to identify some keywords. In biology, recombination refers to the genetic switching of DNA between chromosomes during the process of meiosis in animals, plants, and fungi. A fitness landscape is a 3D model used to represent the relationship between genotype on the horizontal axis and fitness, or how well an individual can survive in a particular environment, which is on the vertical axis. In this model, organisms with a high fitness genotype are seen on a peak, surrounded by genotypes of lesser fitness, and lower fitness genotypes occupy the valleys. Populations will move through this landscape as they experience more mutations. We will see them move up this fitness landscape if their offspring experience mutations that move them around. Fitness landscape with lots of peaks are considered to be what is called rugged, and those with few peaks are smooth. Another word we should know is recombination load, which is an important term to describe the extent to which offspring have a reduced fitness than their parents caused by the separation of favorable allele combinations. The last word is epistasis, which is used to describe how the fitness of an allele can be affected by the genetic background or the other genes present in a particular organism. So how did computer models contribute to the study? Graphs like the fitness landscape were generated by computers based on what the researchers told the computers to generate. In this study, the researchers took four genotypes, A, B, capital A, lowercase a, capital B, and lowercase a, b, and put them on a fitness landscape. The AB genotype represents the most fit individual, while the AB lowercase genotype represents the less fit individual. To some of the groups, they added a capital C gene background and a lowercase c gene background. The physical linkage in model D represents a linkage equilibrium, and model E represents a linkage disequilibrium. A disequilibrium is when the unpaired chromosomes would have the same frequency in a population if the genes were combined at random. So what did they find? The researchers nod at the notion that these computer models suggest populations could evolve to resist creating offspring that are less fit via two ways: reducing the likelihood of recombination events by physically bringing together interacting alleles, or by reducing the strength of the interactions between alleles, making the loci of these alleles more independent of one another. Bringing genes together may be favorable for heritability, and which genes are more likely to be expressed among many individuals. When the physical linkage of genes isn't possible in a population, populations may reduce epistasis with an increase in physical linkage to contribute substantially to mutational robustness, which means how much of an organism's genes stay intact even through mutations. It is important to note that this model has limitations, as we can't really say how each mechanism contributes to real populations found in nature. However, these computer models are good at predicting what could happen when certain conditions are imposed, like an in-lab experiment. Thank you.